right, so first I'd like to just introduce myself. I'm Heidi Baumgartner. I'm the director of ManyBabies, and I have the pleasure of being part of my role is to convene groups of experts like we have here today to bring people together to work on these really exciting collaborations. Just a note, you should have gotten that Zoom warning, but we are recording. And the recording will be made available to the ManyBabies network very soon. Okay, so I'm going to start just by saying a little bit about ManyBabies as an organization, and then I'll hand things off to Mike Frank to talk a little bit more about the project process and expectations of what it means to have a project be part of the ManyBabies umbrella. And then we'll hear from a number of experts in the field of neonatal imitation. And there will be, the presentations will be short, about 10 minutes or so each, followed by a short question and answer period. And then we'll have much more time for Q&A at the end, as well as just a open discussion between our experts. Okay, so just to start out, ManyBabies is a global network. We currently have about 500 people listed on our contributor dashboard. So these are all people who have self-identified, either having participated in a project or that's completed or contributing to a current project. We're working on large-scale conceptual replications and the development of best practices in the field of developmental psychology, with a real focus on open science and putting open science practices into our own projects and encouraging them throughout the field. So there's much more information available on our website. I put the URL there. It's very simple, just manybabies.stanford.edu. Just to give you a very quick overview, I'm not going to go into detail here about the projects that we've completed and have going on. The original ManyBabies project was looking at infants' preference for infant-directed speech. So this was kind of a proof of concept of the large collaboration model. That first project is complete, and there are many follow-up studies, some of which have been completed and some of which are ongoing. I won't get into the details, but just to say that these projects are really a great source, not just of the original project, but they're endless opportunities for spin-offs and follow-up analyses that are really exciting. MB2 is looking at theory of mind in infancy and toddlerhood. They have a stage 1 registered report that has been with an in-principle acceptance, and they are in the final stages of prepping for data collection, as is MB3, which is looking at rule learning in infancy, and MB4, which is looking at social evaluation in infancy. So actually the MB3 stage 1 registered report was officially given an in-principle acceptance yesterday, after a couple back and forths, so that's very exciting. And so all three of those projects are ramping up for data collection, conditions permitting. There's a spin-off of the MB3 project where they're using FNIRS to look at sort of the neuro analog of the behavioral measures that are going to be used in the main MB3 project. So that's very exciting as well. They're working on developing a analysis pipeline. MB5 is going to be examining the Hunter and Ames model of infant attention. They are still currently working on stimulus design and finalizing their stage 1 registered report. And now we have MB6, which is neonatal imitation, and we're just starting that today. We also have a few what we call methodological projects, one of which is called MB Demographics, where it's a group trying to develop a tool for capturing socio-demographic variation across laboratories. They're working on finalizing a manuscript and creating some templates for socio-demographic information collection across contexts. We have ManyBabies at Home, which is looking at online data collection, best practices for collecting data using platforms such as Lookit, and many webcams, which is testing out automatic gaze detection for videos collected via webcam. And those are both collecting pilot data. So just really quickly, all of our projects are expected to follow the key principles of ManyBabies. These are all incredibly important and fundamental to what we do as a network. The first is transparency and openness. So we're committed to being very open and transparent in our processes, as well as open sharing of data and materials when projects are completed. We operate under a model of collective consensus-based governance. So we really value that sort of distributed and consensus-based model. Inclusivity and diversity are very important. So all of our projects are open, and we're always looking for ways to increase the diversity and representativeness of our network. Ethical research is very important. So we have some policies in line regarding like pre-registration and starting and stopping rules for data collection that are independent of results, things like that. And then respect. We expect everyone who participates in a ManyBabies project to treat the others, all of their collaborators, with respect. We have a code of conduct that we've adapted from similar codes used by other groups. The most important things are listed on those bullet points there. This is available on our website. So I won't go into too much detail, but just to say that we take this very seriously. We have a really wonderful community. And so we haven't really had any issues with this, but we want to be proactive and make it clear from the start what the expectations are, including being welcoming and inclusive, respectful of other viewpoints, accepting of constructive criticism, keeping the needs of the community in mind, and showing empathy. All right, and then the last thing that I will mention before handing things off to Mike is that we've adopted the credit taxonomy for contribution reporting across our projects. And this is in line with our goals of transparency and inclusivity. So we ask the contributors to our projects to self-report project contributions in each of these 14 categories that are listed here. So this is an established taxonomy that is being used across the sciences. And project leaders have the sort of leeway to clarify these category descriptions to make them relevant to their project. But this is just an example of a sort of an open science and tool that we're using to across all of our projects. So with that, I will hand it off to Mike to talk a little bit about getting a project started.
Thanks, Heidi. Do you actually mind refreshing the presentation just to reflect?
Oh, yeah.
Get it? Or I can share screen also. It's up to you.
Sure. Let me just stop really quick and get to the right slide.
Oh, here we go. Actually, I'll just do that.
Oh, are you going to do it? Great. That's easier.
Yeah, great. Okay, so you might in part be asking here, why are we talking about using the ManyBabies as an umbrella organization at all to do large-scale projects? And basically our idea in having ManyBabies as the umbrella organization for these projects is that these kinds of big team science projects, large consortium projects, often involve a lot of coordination, a lot of administration, a lot of work, and we don't want folks to reinvent the wheel. We had to learn a lot of things. We're not perfect at these studies, but we've had to learn a lot of things over the course of the last couple of years running projects like this and talking with other organizations like the Psychological Science Accelerator and some of the other ManyLabs projects, ManyDogs, ManyPrimates, and so forth. And so the idea is that we can be a clearinghouse that can help organize projects and bring practices and procedures and policies to these projects to help them move along smoothly, follow kind of existing timelines, templates, and so forth. Further, we can provide some administrative support and encouragement from the perspective of researchers who are interested in this kind of team science. So ManyBabies projects move along kind of with their own leadership team on their own timeline to a certain extent, but then we coordinate as a governing board with the projects. We check in, we get project updates, we coordinate conference submissions to update the community and so forth. So there's a sense of shared goals and shared momentum by being part of ManyBabies. We also have found that there are some analytic and data management challenges that come up in large-scale projects that individual labs often haven't dealt with before. Certainly, I hadn't dealt with those before ManyBabies 1. And as a community, we've now gathered a bunch of expertise in how to deal with these. That doesn't mean we're perfect, but we have some statisticians and folks experienced with data analysis who can help out and answer the call when there are questions about power analysis, modeling, simulation, and so forth, as well as providing some shared tools around data management, data validation, and so forth, which turns out to be a real challenge in large-scale distributed projects. ManyBabies as a network also is a really nice resource for crowdsourcing help for the variety of challenges that come up in infancy research. So we often think about that kind of magical moment that happened in ManyBabies 1 when we were facing this massive pile of data and a tremendous number of inconsistencies and challenges. And we sent out an email, and all of a sudden, grad students and postdocs and faculty from around the world started showing up to our meetings and pitching in and really helping out in sorting out those data, making the data analysis as good as it possibly could be. So the ManyBabies network then is really a resource that projects can call on to try to get some extra hands, extra help from people who really believe in the kind of work that we're doing. Finally, ManyBabies is not a well-funded organization. We're not a really big shop yet, but we are trying to get funds for the umbrella organization. We have some initial support, and we're looking for more. We've also had some projects be successful in acquiring funding, and we can help, we can pool resources, we can try to add letters of support and administrative resources to help you acquire funding to make a project move forward. So ManyBabies 1 move forward on kind of a shoestring, but other projects sometimes require more resources, and concurrent grant applications with an MB project can be very helpful sometimes for adding staff time. Okay, so how will a ManyBabies project proceed? Well, the project starts with a proposal. So in this case, Virginia, Sumeet, and Mark Nielsen initiated the project with a proposal about neonatal imitation. The proposal identifies resources and stakeholders, scopes the problem, justifies why a ManyBabies project would be a good addition to the literature and to the field, and their proposal was approved by the governing board. Now, the proposal is not a plan for every single study and exactly how they're going to go. There's some process that follows from it. Rather, it's the beginning of this broader set of processes that I'm describing. So the next step that we proposed after reading the proposal and getting very enthusiastic about having a neonatal imitation project as part of ManyBabies was to have these workshops that are happening today and tomorrow, depending on what time zone you're in, or today and later today. And so the idea is to engage with stakeholders, to identify key concerns, the things that a ManyBabies project would have to address in order to be a meaningful and impactful contribution. And really just to help catalyze interest in this process, because our idea is that the more folks are excited about a project and the more folks put their eyes on a project, the better the results will be. So we're also looking to form a final leadership team for the project. Of course, the proposers are deeply involved in this, but what we expressed as the governing board is that it was really important to have a broad leadership team as part of this project. Leadership teams are a critical part of ManyBabies projects or any big team science project. The idea is that folks that have day-to-day and week-to-week ownership of the project and push it forward, take responsibility for the deliverables and for coordinating all the different people involved. That's a really key part. Otherwise, projects can founder with a set of responsibility devolving to lots of different people who, of course, have their own individual research projects going on. Leadership teams in general should have a variety of perspectives, a variety of career stages, a variety of backgrounds. These kinds of things can really help move a project forward. So So that's what we're aiming for in this case. So once a leadership team is formed, part of the ManyBabies ethos is an open desire for participation across the community. So there'll be a call for participation to try to be maximally inclusive to folks who are interested in these topics. And the idea being that broad participation at design phases also increases buy-in for data collection. So the more people really have skin in the game, feeling that they own and believe in the design that's going forward, the more they will want to sign up and join the data collection effort, which is often, complicated and costly. But if they really think that this is the right study and have looked at the materials and understand it and believe in it, then they're more likely to devote their resources in their individual labs to collecting data. Finally, and this is really the heart of the process, is a series of design meetings around what the study should be. As Heidi alluded to, we've taken full advantage of the idea of a registered report publication process for ManyBabies. This is where, of course, you develop the idea, design the study, and then submit a stage 1 registered report for review prior to data collection. This extensive planning and manuscript discussion process really helps with ManyBabies projects for a lot of reasons. First, peer review can help us catch issues that would potentially compromise the study or decrease its value if we carried it out without review. So in a number of cases, peer reviewers have identified weaknesses that we've then addressed, and that really maximizes the value for the effort that everybody's going to put in. So because the stakes are relatively high, we really want to get it right. Second, the submission of a registered report really acts as a very strong pre-registration, making sure that we have everything sorted out before we begin data collection, especially on the analytics side and the data management side. And third, the registered report also anchors the contribution. As you saw from the timeline, these projects take many, many years. And so it's important for especially the early career researchers who are involved to have something to show for their efforts sooner than when the final project is done, which is often after they're finished with graduate school or after they're finished with their postdoc. So this publication process really works for us. And what leads up to that in the ManyBabies project is an iterative process of consensus-based concept refinement. Really, you get together and try to hash out the details of what the right study would look like. So we try not to be bound by prior work, in the sense that what we want to do is the best possible study to make progress on a particular question of interest or on a particular phenomenon of interest. And then once the study design is reached, the leadership team then can convene subgroups on stimuli, on analysis, and so forth to try to really get the details worked out and to draft the relevant parts of the registered report, for example, the analysis plan or the specific procedure. Another hope for this part of the process is that even just by planning the study, you create a set of reusable procedures and materials and analytic standards that then can guide others as they work on these questions. In ManyBabies 1, we found that was certainly true. I mean, just the process of discussing, okay, what's the right attention getter to use for this study actually resulted in a bunch of folks changing the attention getters they used in their labs. So really the process of discussing our craft and what we do and how to create the best possible study can really be a very useful thing. All right, so that's basically what we are hoping will happen from this point forward. And I just want to end by reiterating our dream here, the reason why we're doing ManyBabies projects and the reason why we've been interested in this organization. The dream is of a cumulative developmental science, the idea that we can build on one another's work to really advance our understanding of infancy and child development, and where we work together to make precise measurements of the things we care about, to create shared resources, develop best practices that shape the field and guide the field going forward, and resolve some of the longstanding debates that have plagued our field. And that we can do this in a collaborative and inclusive community that moves forward by group discussion. Those discussions aren't always easy. They're often challenging, but they're incredibly important. And working through these issues often, for me, and I think for many others who've expressed this, results in a lot of growth and understanding and forward progress. So that's it for what I wanted to say. Thanks so much for everybody for being here and for participating in this process. Thanks to Virginia, Sumeet, Virginia, and Mark for initiating this project. And I'll turn things back over to Heidi.
Great, thanks, Mike. So we're going to move on to the presentation portion of the program. Before we hear about the proposal from Sumeet, Virginia is going to say a few words just about sort of how this all got started. So I'll hand things over to Virginia.
Thanks, Heidi. Actually, what I wanted to do was introduce Sumeet, because the rest of us panelists have been reading and writing about each other's work for some time, but you probably don't know Sumeet. So I want to explain. Sumeet has graduated with a PhD from McMaster University, where his research was on automatic imitation in adults. And then last year in November, he moved over to work with me on another imitation project here at the University of Queensland. So when we were approached to do this proposal, Sumeet took the lead, and that was really quite deliberate because he comes with absolutely fresh eyes. He wasn't trained in development. He didn't know the neonatal imitation literature until he started reading it very carefully. And so this proposal that you're about to hear about, it's really from a clean perspective. And I think that's one of the strengths of it. Obviously, it's going to change as we talk through it. But I think it's nice that we start with somebody who doesn't already have established opinions on this phenomenon. So that's what I wanted to explain.
All right, great. Thank you. Yeah, I think that is really interesting context. So I appreciate that. All right, so now I'm going to, Sumeet has recorded a presentation, so to make it easy for us to share with the next workshop as well. So I'm going to play that, and then we'll have a few minutes if people have sort of clarifying questions before we move on to the other presentations. So let me do this.
Get into the proposed project, we would first just like to acknowledge
Okay,
the
thanks,
traditional
everyone.
custodians of the land where we work and live, and we'd also like to pay respect to the elders past, present, and emerging. There is no question that humans show an overwhelming proclivity for copying others, which underpins cultural learning and contributes to the enormous diversity of behavioral traditions observed across human groups. However, there is still a significant amount of debate on the question of whether newborns imitate the actions of others shortly after birth. As such, our team at the University of Queensland was approached by the ManyBabies initiative to come up with a project in collaboration with other experts in the field to address this controversial issue. Before we get into any of the project details, however, we just want to provide everyone with an overview on the current views on neonatal imitation and also discuss the existing criticisms used against papers that have found no evidence of this phenomenon. After that, we're going to wrap everything together and introduce you to our project proposal. Currently, there seems to be a divide between researchers who have found evidence for neonatal imitation and those who have not. The first finding that imitation is present from birth was reported in Meltzoff and Moore's seminal work in 1977. They found that infants between 12 and 21 days of age can imitate both facial and manual gestures. This seminal paper has close to around 5,000 Google Scholar citations, and the central claim that newborns can imitate has permeated multiple subdisciplines of psychology and has taken hold in adjacent fields including medicine, philosophy, and parenting. After this initial paper was published, the findings were confirmed and extended in a variety of independent laboratories since 1977 in reputable journals such as Society for Research in Child Development, Science, Infant Mental Health, and Developmental Psychology. Unfortunately, despite its intuitive and theoretical appeal, the issue of neonatal imitation was far from settled. Within published studies, findings were mixed. Some report replications of the original findings and extend the range of gestures imitated, while others report imitation of just a subset of gestures. To make matters even more complicated, Other independent laboratories have reported no evidence for neonatal imitation in reputable journals such as Developmental Psychology, Infant Behavior and Development, Current Biology, and Infancy. In order to address these mixed findings in the literature, researchers in favor of neonatal imitation have put forth some criticisms pertaining to the experimental design and procedures used in studies that have challenged the existence of this phenomenon. However, before we go ahead and discuss them, there are a few elements of experimental design both sides can agree with. First, researchers from both sides agree that this phenomenon can be captured at the behavioral level, indexed by frequency of imitative behavior and/or duration of imitation. They also agree that the best way to assess imitation in newborns is by using the cross-target comparison method, which allows researchers to directly compare the frequency of imitative behavior during action observation to the frequency of the same behavior to the other gestures, also known as control gestures. And finally, in most newborn studies, the stimuli is presented typically at a distance of 20 to 25 centimeters from the infant's eyes. With that said, there seems to be a lot more differences than similarities amongst researchers from both sides in terms of methodology. These differences have been highlighted by those in favor of neonatal imitation. For example, recent work that has challenged the existence of neonatal imitation has been criticized for not carefully considering the arousal state of infants during action observation sessions. Many researchers have argued that the null findings reported may have simply been due to the infants being in a drowsy state, which would have undermined their ability to process visual stimuli. Other work that has challenged the existence of neonatal imitation has also been criticized for using inadequate stimulus presentation periods during testing sessions. Indeed, it's been argued that short presentation periods may undermine neonatal imitation due to the fact that infants require additional time to process visual information or fixate on a stimulus. In addition, researchers in favor of neonatal imitation have also argued that the null findings reported by more recent work may be due to short response periods as well, which don't consider the fact that neonates may require more time to organize their motor responses to visual stimuli compared to older participants. Previous work has also been criticized for not providing infants with adequate postural support during action observation sessions. Indeed, it's been argued that postural imbalance can be disruptive to young infants and their actions. Work that has challenged the existence of neonatal imitation has also been criticized for having poor control over maternal leave-taking during the testing phase. Indeed, these researchers have argued that the immediate presence of the mother during the testing phase may actually dampen imitative responding in the infants. In addition, although imitation has been traditionally coded when an individual is meaningfully engaged with another during social interaction, some researchers have argued that it's equally important to code potential imitative behavior when the infant has disengaged from the model. Indeed, these researchers suggest that some infants may look away from the model in order to adequately process visual information or organize a motor response. And last but not least, some work has also been criticized for not controlling for model familiarity during testing sessions. Due to the ongoing controversy pertaining to neonatal imitation, our team at the University of Queensland was approached by the ManyBabies initiative to formulate a project proposal to address this controversial issue, subject to feedback from experts all over the globe. The main goal of ManyBabies is to bring labs together to address outstanding theoretical and methodological questions about the nature of early development and how it's studied. Now we're going to be moving on to the final section of today's presentation, which is our ManyBabies project proposal. Please note that we offer it as a starting point for collaborative decision making. As mentioned earlier, infant imitation has traditionally been examined using the cross-target comparison method. We would like to use a similar protocol for our proposed ManyBabies project, but with some important modifications, which will be discussed below. Although it may seem optimistic, we would like each lab to recruit 60 infants that are 2 weeks of age and another 60 infants that are around 6 weeks of age, leading to a total of 120 participants. We would also like to explore the following gestures: tongue protrusion, mouth opening, and sequential finger movement, as these have been observed in several studies. We would like each lab to code behavior based on the guidelines used in work favoring neonatal imitation, and in order to address previous criticisms in the field, we would also like to ensure that each lab code potential imitative behavior even when the infant is disengaged from the model. In order to simplify the data collection process for this project and eliminate any concerns about the potential for significant attrition or caregivers practicing gestures with their infants between visits, we suggest using a cross-sectional design. In addition, in order to reduce systemic variation and complexity of data collection, it's been proposed that mothers bring their infant to the lab instead of having experimenters complete in-home visits for data collection. We also think it would be best to conduct research data collection in a testing room with one desk, a standardized infancy with no other distractions. Since some work has also found evidence for neonatal imitation using video stimuli, we thought it would be nice to add a video stimuli presentation component in addition to the live sessions in this project. As mentioned earlier, work that has challenged the existence of neonatal imitation has been criticized in a variety of ways. As such, for our project, we will adopt a recommended procedure that will sufficiently address each of these concerns. First, in order to address the issue of infant arousal, we will only include infants that are in state 4, which is quiet alert, or state 5, which is active alert, as part of our study. In order to address the issue of response periods, we will be using the recommended suggestion of 2.5 minutes for each gesture. In addition, as recommended by researchers in the field, each gesture will be presented for 90 seconds in a burst pause pattern of 15 seconds passive and 15 seconds active. In order to address concerns about postural imbalance, we recommend that each lab have the same infant seat for testing sessions. As mentioned earlier, work that has challenged the existence of neonatal imitation has also been criticized for having poor control over maternal leave taking. As such, we will ensure that the mother will not be in the line of sight for the infant. And last but not least, in order to address concerns about model familiarity, we will ensure that the live model will only appear during the testing phase. In addition, for the video presentation, the model will be different as well. Finally, in the spirit of open science, effort will be made to publish testing session videos to Databrary, a restricted access data library that is specialized for storing and sharing audiovisual recordings collected as research data. One major goal of this platform is to increase scientific transparency in order to deepen insights and accelerate discovery in developmental science. We have also created a proposed timeline for important elements of this project, but of course this is subject to change based on your feedback. And that's all for today's workshop. Thank you so much for listening, and we look forward to hearing your feedback.
All right, great. Yes, I saw a lot of hand clapping there, mute hand clapping, but I just want to say thank you so much, Sumeet, for recording that great presentation. I think we can all appreciate all the thought that went into, sort of compiling the information that's out there and thinking through what a good starting point would be. And also, I just want to reiterate, as Sumeet did in his video, that this is just merely a starting point for our discussions, just something to get everyone thinking in the same direction. So with that, I'll open up the floor to Virginia and Sumeet. If anyone, I know we'll have time for more discussion, but if anybody has any quick clarifications, we could take time for a question or two from the panelists. Does anyone have anything that they'd like to? All right, I think, yeah, let's just continue with the program then. So next, oh, Mike, go ahead.
Let me just mention, one of the sort of spirits of ManyBabies chaos and interaction is that, sometimes we want to move along with the Zoom proceedings, the auditory proceedings, but if you have comments that you'd like to note or discussion that you'd like to do, you can use the Google Doc that Heidi shared. That could be a great place to compile the sorts of questions and concerns and comments that we bring forward. I've already started writing some notes there, and I think others have as well. So yeah, let's move forward, but please do engage over there.
Great point. And that document, I think that is available to everyone in the audience as well. So everyone is welcome to contribute in the spirit of ManyBabies. So with that, I'm going to hand things over to Andy Meltzoff. We're really lucky to have, it's not always the case in projects like this that you get someone who was involved in developing the whole area of research to offer his experience and expertise. So I'd like to hand things off to Andy.
Good, thanks Heidi. And Sumeet, thank you for that presentation. Welcome to the community. I'm sure we'll have a lot of time to be able to discuss things over the next several months and years. That was a very good presentation. Thank you for doing that. And thank you, Michael and Heidi, for inviting me. I'm happy to be here today. It's obviously a topic that I care deeply about. And I think it will be exciting and productive to have this be a ManyBabies project. So I was thrilled that the board picked it and that we can benefit from some of the learning that ManyBabies has already done and the resources they can bring to this. It certainly could not have been done in the way it's likely to be done if it were just labs privately connecting with one another to coordinate with 2 or 3 labs. So thanks for doing that. I also think that the project's possibly going to be important for psychology and developmental psychology in particular, because it's a topic that has been discussed for many years. And there are issues about how to have different labs use the same procedures. And I think that's sort of what ManyBabies is built to solve. So it seems a particularly productive place to start this project. Now, I will note that even though ManyBabies has tried to standardize procedures across labs, that remains a challenge. It's something that I hope we can dive into deeply here. Even in the infant-directed speech first paper, there were reported interlab differences. And so that's something we're going to want to keep our eye on as we develop procedures that we can use. And I have a couple suggestions about that. One of them is that perhaps we can, this is all going to be part of group discussion, obviously, but I would like to put it on the table. After the dialogue and the agreed upon standardized procedures, we might find it helpful to do a joint or a group pilot study of, obviously that's quarantined from the real start of the experiment. We wouldn't make any confusion there. But the idea would be, in view of the past ManyBabies studies where there still are interlab differences and differences in procedure that make a difference, in neonatal imitation and work with very young babies, that's exaggerated many-fold. So I would like to propose we get our standardized procedure, and then we have a phase where everybody videotapes while we're using that procedure to test a certain number of pilot studies in each lab, circulate the videotapes, and that will allow everybody to see what the other labs are doing. And if one lab is inadvertently deviating from the agreed upon procedure, we can catch it early and make the changes there, because I think they could be very important. So that's one suggestion. Another suggestion is perhaps, again, we'll talk about this, we could use one age and keep the design simple to begin with, just to begin with. And here, if that's what the group decides to do, I would suggest using 6-week-olds because they have much better posture than 2-week-olds. Their state is far easier to maintain. They're plenty young so that the core question can be addressed. And if it can be found, if across labs we can elicit imitation in 6-week-olds, if across labs, then of course what we'd want to go do is go younger. We can go down to 2-week-olds or do what I did of go down to newborns in a hospital setting. But going to hospital setting is probably not the right place to start, as suggested by the proposal. So my tentative one is let's find in a very early age, but one where the children are not so fragile and are not so able to be startled, drowsy, fall asleep, hard to keep in a good state, and so forth. A second suggestion is that we might start with the best, with a simple design and a clean design. And in our lab and others around the world, we have found that there's persistence in the response, sometimes called a carryover response, that works like this. If you show tongue protrusion in the first block of gestures and then switch to mouth opening or switch to another gesture, another one after that, there's also often motor persistence, sometimes called carryover. And the interpretations of that range from just simple repetitive behavior on the child's part to some sort of social reason for it. But the reasons don't matter. What does matter is that people have found carryover. And so trying to do an independent groups design, which I think I heard Sumeet suggest, is probably a good idea. And I have done independent groups design where we've used, in 6-week-olds, we used tongue protrusion for one group. And what we did is test babies on 3 successive days, which I found to be particularly powerful because it was able to get a more stable reading on any one baby. Instead of one shot for 2 minutes, 2.5 minutes, you can test the baby on 3 successive days. And our results were really very powerful for that in terms of eliciting imitation. It may be because you can test the children for longer when you have 3 days in a row. It may be because it approximates something like naturalistic social interaction when the child re-encounters the same person one time, another time, another time. I think the powerful part is the baby only sees this person do one gesture and there's not as much confusion on the baby's part. Now I'd like to maybe switch in the interest of time and just talk generally about 3 categories of things that I think of as moving in the direction in a non-artifactual way of being a key to the study. What are the most sensitive non-artifactual conditions, sensitive eliciting conditions for babies this young? And I'm only going to give examples of this. There's 3 categories of things I can sort of just use as examples here today. The first is general experimental context. And here I note just a couple of things. In none of our studies did we tell the parents that we were testing facial imitation. Way back in pilot studies a long time ago, I found that if you told parents you want to test facial gestures like tongue protrusion, parents would specifically practice those behaviors at home, and it would interfere with the experimental protocol. A second thing that fits within general experimental context is that we used very, very dim lights. The room lights were often turned off, and as specified and talked about in the article, we have a spotlight behind the baby seat that's shining on the adult's face and a completely homogeneous background behind the adult. And we have found that this helps, obviously, focus the attention of the baby on the stimulus, which is the moving face, rather than looking around, and you can maintain infant attention using that protocol. The second category of thing I want to talk about are maybe baby factors, having talked about general context. And one was, I think, mentioned by Sumeet, but I'll mention it again here. In none of our studies have we shown the experimenter's face to the baby before the moment that the experiment starts. We want to keep the experimenter's face novel under these experimental conditions. Now, that's not a claim that babies don't imitate their mother who they live with every day, re-encounter every day, and mothers and babies play particular facial games. But we're trying to do this in an experiment where we have to start at a particular time and do a protocol. And we have found out that it cuts down variance to just have the baby encounter that face as part of the experiment rather than doing random things beforehand. Also, the state of the baby is very important, as has been mentioned. And when we have smaller working groups, we can talk about techniques and ways of maintaining or starting the experiment with the baby in a calm, alert state. The fact that they're calm and alert and not overly active, eyes wide open before this protocol is going to start, it's going to run off according to a clock. You can't stop. So the baby needs to be in a very excellent state to start with. Third class of things I want to talk about was procedural issues. And we developed early on this burst pause pattern. And I think that's a very effective and important way. I'm glad to see that Sumeet and the group is planning to use that. Scoring also is very important. We need to have good definitions of what counts as a tongue protrusion and what counts as a mouth opening. In closing here, because my time's running out, as everybody knows, some things in developmental psychology are much easier to elicit than others. Infant preference for infant-directed speech should be one of the very easiest things to demonstrate. And it's interesting that in that first paper, there were differences, variations in effect sizes due to different inclusion criteria on the babies and different techniques used in different labs. So even something as potentially robust as just preference for infant-directed speech depends a lot on eliciting conditions. I think that's particularly true in imitation in this extraordinarily young period. 6-week-olds are probably the youngest kids, or 6 weeks and under, that ManyBabies has ever had in a project. And one will see that there's lots of techniques that have to be used with them. So that's one reason, again, that I wanted to emphasize the pilot in closing here, to make sure that the general experimental context, the baby-centered factors, and the procedural issues are all really and truly standardized across labs, and people have had a chance to discuss the motivation for particular characteristic and particular features, procedures, so they don't seem to come out of the blue, but make sense about why they're being used. So that's it. Thank you.
All right. Outstanding. Thank you very much, Andy. I think I was seeing a lot of people taking notes during that. Certainly lots there for discussion. If any of the panelists would like to have any reactions to that, or would like to ask a question before we move on to the next speaker, we can do that now. All right. I think let's just keep it moving then. Next, we're going to see a presentation from Kathleen, Akins, and Nazim, Keven, who are both here today, but we've got a recorded presentation just because we were unsure about timelines and such, or time zones, and who was going to be able to make it. So I'll play that, and then we'll have time for questions after. Hopefully this will go a little more smoothly this time.
First of all,
Do we have audio?
thank you very much for inviting Nazim and I to this workshop. We were really, really pleased to be invited. As many of you know, Nazim and I are both philosophers. Both of us have neuroscience backgrounds. And as you also probably know, most philosophers are what you might call natural contrarians. And we are hoping that even as philosophers, we can make a sort of a productive contribution to this workshop, although from a bit of a different point of view. Now, it was Nazim who first became interested in neonatal imitation when he was an MA student at Fraser University, and I was his advisor. And after reading everything there was to read at that point in time on neonatal imitation, this thorny question just kept arising. And that thorny question was something like, good God, why would it be tongue protrusion? Now, what we meant was, you know, out of all the possible gestures that one could imitate, why would tongue protrusion work? And that got us very interested in understanding both the origins and the function of exactly the sort of behaviors that were being tested in the imitation experiments. Now, eventually, after a lot of looking at research in the neurophysiology of fetal and neonate development, what we came to was really, although we didn't know it at the time, was an extension of Esther Thalen's theory of rhythmic movements, or stereotypies. Now, stereotypies is really sort of a terrible name for this, given that these rhythmic movements, every time an infant produces one, it's always somewhat different from the last. And in fact, that characterizes them, the fact that they aren't just sort of repetitive movements. Now, Thalen in 1979 reported in her famous article that she had followed 20 neurotypical infants, and she recorded every 2 weeks from 4 weeks to 1 year. And this resulted in 16,000 recorded instances of rhythmic movements. And on analysis, what she found was first that the peak postnatal frequency for each type of stereotypy was determined by anatomy. So, for example, all stereotypies involving the legs, such as kicking with alternate legs or synchronous heel thumping, my favorite activity, that these things peak, all of the leg activities, at 20 weeks postpartum. And the second conclusion was that 84% of the stereotypies recorded had identifiable releasers, such as the appearance of the caregiver, the presentation of a toy, or say an interruption to feeding. Yet these releasers were remarkably non-specific and unrelated to the rhythmic behaviors they listed. So what we did was to look backwards from 4 weeks postpartum to see how those behaviors arose in utero and to try to understand what they were functionally. Now, we came to sort of 3 basic conclusions, and they were these. So looking at the ultrasound images, one sees a set of individual aerodigestive rhythmic behaviors, starting at about 9 weeks post-conception. And this begins with a very rudimentary swallow. Now, more of these stereotypies are added thereafter. Almost all of the rhythmic movements turned out to be components of aerodigestive motor runs of the kind that would be needed at birth. So through rhythmic repetition over and over and over and over and over, various bouts of rhythmic repetition, protocomponents of the aerodigestive behaviors that first emerge then begin to coalesce into these primitive, rather jerky motor sequences. And then with more repetition, they evolve into these smooth, tightly coupled motor runs by 32 weeks post-conception. So in other words, by 32 weeks, infants are good to go, or at least as good as they're going to be to go, in terms of the basic aerodigestive functions of suckling, swallowing, breathing, and so on. Now, Thalen concluded, as did we, that the ubiquity of these stereotypies and their order of appearance in mammalian development, they're not the only ones, suggests that these constitute a functional stage in sensory motor development. And in particular, given the predominance of activity-dependent cortical development at that stage, it seems likely that these states serve a neurodevelopment function. At birth, sensory motor maps of the body in cortex, in M1 and S1, they exist, but they're not functional. One neuron does not connect with and communicate with the other. And so thus, sort of prior to the advent of a functional cortical spinal tract for voluntary movement, and this occurs at about 4 months after birth or begins, sensory feedback from infant stereotypies likely plays a role in developing those requisite cortical connections for voluntary movement. And this is very much in line with the way in which these body maps begin to be formed in utero, namely through activity-dependent development. Now, somewhat suspiciously, when we looked at the timelines for all of these individual aerodigestive rhythmic movements, each of them lines up with, in the beginning and for the time at which they're eventually extinguished, with the timelines that have been given in the neonatal imitation literature. And this led us to ask whether what we're seeing during imitation experiments is indeed something over and above these bouts of stereotypies, which presumably are tempered by context and whatever other factors affect arousal. Now, this worry was deepened by the fact that, or when we learned that, within the first 15 minutes after birth, 95% of all full-term newborns make spontaneous tongue protrusions, and almost all of them occur within the first 3 minutes after birth. Now, this suggested to us that there was a very strong tie between autonomic function and between tongue protrusion and the other aerodigestive rhythmic behaviors. So it seemed to us that there were these two hypotheses now. One was the imitation of oral facial gestures, and the other was a theory of arousal-dependent aerodigestive stereotypies. And the question that we would love to be able to answer is really this, which is, if one were to design an experiment today, how could we distinguish experimentally between these two hypotheses? What one has to show in order to infer that behavioral response is evidence of imitation is that these gestures do not arise purely as a function of aerodigestive development and arousal. Now, arousal is tricky. From the onset of the first stereotypies in utero to their extinction after about 1 year of age, the physiology of arousal is rapidly changing. And indeed, prior to the emergence of sleep-wake cycles in utero, which at the beginning there simply aren't, arousal is actually defined by the onset of these stereotypies. That is to say, a particular bout of rhythmic behavior lasting 23 seconds, etc. That's what constitutes arousal. And at birth, arousal seems to drive these rhythmic movements. The problem is that, as Phelan reported, stereotypies quickly become prevalent during what she called non-alert states of the infant, a trend that really peaks at about 20 to 26 weeks after birth. In other words, you're getting what seems to be a complete reversal in the relation between arousal and these bouts of rhythmic behavior. Now, we think that this change makes sense because it likely represents sort of a changing landscape of infant motor control from subcortical oscillators at the outset and to cortical control of voluntary behavior. And obviously at 20 to 26 weeks, that is the period at which we start to get the onset of voluntary behavior. Now we have two suggestions, really. You know, all of this makes it incredibly difficult to know how to separate the effects of arousal and imitation in the experimental context. Fortunately, research on the neuroscience of infant autonomic function has seen a huge boost lately based upon a big interest in the development of emotions. That said, we've yet to do any real investigation into the relation between arousal and bouts of this orofacial activity. And this leaves many questions unanswered. For example, is the relation simply scalar at birth? Does greater arousal lead to a greater incidence of these rhythmic behaviors? For another, what sorts of external stimuli precipitate arousal and hence these bouts of behavior? Do different levels of arousal alter the incidence of particular rhythmic behaviors? For example, in low arousal, do you have a greater incidence of, say, mouth opening to tongue protrusion? And at high arousal, you have a greater incidence of, of tongue protrusion to mouth opening. This is entirely possible. We just don't know the answer. Another question, what is the refractory period of an arousing stimulus, such as the presence of the experimenter? If you were giving a sequence of stimuli, are you getting refractory effects between presentations? And I think that's the sort of thing that everyone would want to know in conducting an experiment for neonatal imitation. It would really help us to design experiments where we could control for arousal and see whether there was additional behavior that could be attributed to imitation. In any event, if we had these sort of answers in hand and we had these kinds of control experiments, I think it would be absolutely necessary that all infants should be monitored for arousal from the moment that they were seated through the end of the experiment. And then later, one could check statistically for the influence of arousal on the data collected in each individual case. I think that if we were to do these two things, it would go a long way to disentangling the effects of arousal and imitation. And in particular, it would sort of give us agreed upon guidelines for how we're supposed to conduct the kind of good experiments that both sides of the debate really hope that we conduct. Okay, and that is the end. Thank you very much.
Okay, great. Thank you so much. That another great presentation. You're starting to see some common themes as well as some new avenues for discussion. Would anyone like to ask Kathleen or Nazim any questions while we have them here? All right, great. We'll just keep moving then. I'm going to hand things off next to.
Sorry, Heidi, could I just jump in there?
Sure,
So
please.
Is the plan to save our discussion questions for the end once we've all kind of given our presentations? Because I think that might be more productive because there's going to be a lot of common.
No, I was just wondering if anybody had any quick clarification questions or something that they didn't want to let too much time pass between the presentation, but yeah, we will have plenty of time for discussion.
Okay, fantastic. Thanks very much. Cheers.
Okay, great. So with that said, I'm going to hand things off next to Laurie Bayet from, I'm probably saying that wrong, from American University, who I very luckily had the opportunity to meet in person just this past weekend while we were standing in line at the coffee shop at the ICAS conference. And then I got to see her give a talk at the award ceremony that happened right after that. So I'm going to go ahead and hand things off to her. I'll put the spotlight on here. Take it away, Laurie.
All right, can you all see that, the slides, hopefully? Awesome. All right, so this is going to be a little different. So disclaimer to start, unlike a lot of great people in this Zoom room, I'm not a neonatal mimicry expert. I'm an infant vision and face perception researcher. But as I'm sure you're aware, right, neonatal mimicry has important implications, I think, right, for the theories about how we think the infant kind of sees the world starting and other people's faces starting from birth. But also because I'm not a neonatal mimicry researcher to start, even though I'm interested in this, in kind of knowing what the answer is, right, to this question, because of the implication for vision, the work I'm going to present, it would literally not have been possible without the critical contributions of Elizabeth Simpson, who is on this call. That's starting from the very, very start of this work. All right, so with that, all right, so the motivation for that work was to kind of take a look at neonatal mimicry. Obviously, everyone in this Zoom call agrees that this is an important question to settle. And the approach we took was to use the open source Lookit platform, which you may have heard about, which is this online platform for studies of infants, with the idea that having that design would allow to have kind of a scalable procedure that could be applied in many different labs, even in settings, potentially producing larger samples that what can be done in the lab, and also kind of promoting that transparency of methods, right? That we know from meta-analysis and so on, all that work, that methodological differences between labs can be a driver of differences in findings. So that was part of our motivation. Why doing this online? So here, the idea for this was that it's part of the reason that we think part of the reason that neonatal mimicry is still, there's still a lot of debate on this, is just because it's incredibly hard to study, right? And it's incredibly hard to study newborns, and part of this is that it's hard to reach newborns, have them come to the lab. And so hopefully bringing the lab to them, right, by having that asynchronous online platform would facilitate that. So that was the rationale for it. And finally, we also took a slightly different approach, right? While this, the ManyBabies project is focused on imitation of experimenter movements, we actually wanted to focus on caregiver movements, right? Which there's lots of reasons to think that actually may not even happen at all, right? Whatever you think about imitation or experimenter. So that's a very important caveat for everything I'm going to talk about, that we're actually trying to answer a slightly different question in this work. But the reason we're doing this is because whether or not neonatal mimicry can happen, right, in controlled conditions, I think whether or not it happens with caregivers is important for understanding how infants, newborns learn in terms of visual learning, social learning in the first few months. So that was our motivation. So essentially, our first goal wanted to see if it was feasible to ask this sort of question using an online asynchronous platform, Lookit, because we were hoping that the online platform would allow us to reach a slightly more diverse sample than it's typical in the lab. We also wanted to look at the diversity of our pilot sample. And finally, from that pilot sample, again, not powered to get robust estimates, but we still wanted to get preliminary answers on neonatal mimicry of caregiver facial gestures. And we also asked caregivers to judge mimicry in that study. All right, so with that, so here a little aside on kind of meta discussion, but this is I went back to my email and looked, and the first email discussion on this, so it was in November 2017 with Kim Scott, then at Lookit. So it's been a while, but we did completed usability piloting in 2020. And then from that, we were kind of refined the panel a little bit, and we've completed a collection of our feasibility pilot, just 29 participants, new participants, right, that hadn't been recruited before, over about 6 to 16 weeks. And so they were compensated. They could participate again up to once a day if they were in the eligibility window, which was 0 to 42 days of age. And importantly, this was a between participant design. So they were randomized to having caregivers model either tongue protrusion or mouth opening. And they stayed in the same condition if they were participating again multiple times, which you'll see some of them did, some of them didn't. And so how did we make this work? And another important point is that they could restart the test trail if the child became fussy, right? Which happened, it's a fair part of the time. Say they start the test trail and it turns out, the infant starts crying and they could start again. And so here, one point that may become important later is that, and I think there are other reasons also, if you think that the baseline gesture rates, which we did use as planned in analysis, in some analysis, may not actually, may not be interpretable in the same way that those in the labs would be for many reasons, including this is a caregiver, but also if they started the test trail again, we did use the earlier baseline period that we had. However, they may still have some repeat going on. So just bearing that in mind whenever analysis use baseline rates. So how did caregivers know what gestures to do? They were guided by auditory cues that sounded like this.
Continue looking at your baby, but make a neutral face.
This is where I realized that I mean I have checked the box to share the sound. So could you hear that? Or do I need to start showing it? Okay, excellent. All right. So essentially they were guided by cues, so they kind of knew what they were doing at any point, and we used a classic verse pose design, 1/82. All right, so the next step is once we had all those videos, right, was to kind of process those videos. And this was fairly time intensive, but essentially we had 2 raw videos from that work. One was the practice video. So before the test trial, we had caregivers practice the gestures just to kind of essentially so that we could check that they were doing the right one. And then the test trial video, right? And so here, importantly, we had one person handling video processing in 2 ways. So one, they were blurring the video in one way where the caregiver face was blurred. And all of the condition-specific task cues, right, like stick out your tongue, were edited out so that we could have quote unquote blind coding, right? Second, the same video was processed in a different way with the newborn face blurred, and we use this for quality checks. And so when I say quality checks, is to just 2 things. So one, does the video look like it can be coded properly, right? So no video lags. We're dealing with webcam videos, right? So there could be a lot of different things that could happen. And also just, is the caregiver doing the task correctly? And then we had separate people handle coding of neonatal gestures, again, blind to condition with criteria similar to lab protocols. So this is, again, critical contribution with Lynn Simpson, right? All of that training of coders and so on happened in this way. But coders were able to code those videos reliably, which is important for us. And so here, really manual processing, as I'm sure everyone is aware, was extremely time intensive and required a lot of different people, lots of training. And so I think I would really concur with the notion that if some of this could be, at least if there could be a parallel stream of coding with automated coding, that could really push that sort of effort forward, make it more scalable. All right, so how did this look like? So we were talking about repeat participation, right? So here we had 29 different people. About half of them, almost half of them participated more than one time. So each dot on a horizontal line is a different participation by the same infant. And if the center is green, that's a video that ended up being included. Overall, one thing that we did not anticipate is that some caregivers just held a gesture during the modeling period instead of repeating it, so we had to exclude those. But overall, about a bit more than half participants managed to have at least one video included, so 55%. That's pretty standard, I would say, for online data collection, particularly with this sort of age range. Okay, so that's for the feasibility, right? Overall, it looked like we were able to collect this sort of data with the same sort of success rate, attrition rate that we would expect for an online study. In terms of participant demographics diversity, here I'm going to go a little quickly, but I think because I'm not sure it's super relevant to this audience, but essentially we were able to have a sample that was relatively diverse in a number of criteria. So one, we were focusing on the US, right? We had people from different states. Most caregivers who participated identified as women. In terms of the kind of race and ethnicity of their families, we had some diversity there, as well as in terms of setting, rural, suburban, urban. We looked also for people for caregivers who identified as birth parents at their postpartum depression scores, just to get a sense of how we were doing in terms of representativeness for postpartum depression. And roughly speaking, I think we're actually in line with Sumeet's prevalence, right? So we had about 17% that screened above some cutoff, clinical cutoff. So overall, it looked like what we would expect in this population in terms of our depression scores. Now, the one thing that we did quite, there's a lot of things to improve there was in terms of caregiver education. Even though we were recruiting on things like Facebook, Instagram, we vastly underrepresented caregivers with less than college education. So just something to focus on kind of in further recruitment efforts. Income was less marked. I don't think it was significant. We still see a trend, but for education, it was just really obvious and glaring. Okay. All right, so what did we find? And so here again, this is mimicry of caregiver gestures, right? But here is what we found, again, using types of analysis that you would typically use in lab studies. So the first thing we did, because we had those repeat sessions, is we can average the gesture rates So how many mouth openings, how many tongue protrusions newborns were doing per minute in each of those sessions, and average it. If say a newborn had 6 different sessions included, we can average that, get a more robust estimate. And what we found from this is that essentially we didn't see a difference in gesture rates based on the condition. So based on if the caregiver was randomized to middle mouth opening or tongue protrusion, it kind of looked the same. And that's whether if we looked at the raw rates of gestures during the test period, so either modeling or rest, right? But after that initial baseline, or if we correct for the rates measured at baseline, again with the caveats on what baseline means, since we're talking about a caregiver participating multiple times, right? Right, but either way, we didn't see a condition difference there. Now, and that's for 16 included participants in that feasibility pilot. Now, we also looked at a different session-based definition, right? So sometimes in lab studies, right, people will say, well, this is an imitator, this is not an imitator, based on if an infant has a positive increase in the matching gesture during a test from baseline, and this increase is larger than for non-matching gesture, right? So here, important caveat, this does use the baseline rate, right? So just bearing that in mind. Now, what we could look at, so now we have the 66 sessions from different people, different infants, right? And we can compare the proportion of sessions that end up being defined as imitator session, right, or mimicry session, based on to an empirical nodal distribution, right? So if you randomly shuffle the condition for all of the sessions, then we get kind of a distribution of which sessions get labeled as what proportion of the sessions get labeled as imitator or non-imitator based on that shuffling of condition. And if we do this, and if we compare our actual experimental proportion of sessions that do end up labeled as imitator or not, then we find that we don't get more sessions labeled as mimicry than expected by chance, meaning by just randomly shuffling labels and doing the same sort of analysis. So essentially at the session level, again, at the group level, we don't see evidence of mimicry, with, again, the caveat that this procedure does use the baseline rate. Now, finally, a last thing that we did that isn't, I would say, standard for the field, but perhaps can inform future efforts, was we also ask caregivers, right, whether they think their newborns were actually mimicking them, because we're interested in that kind of ecological question, right, of if neonates can mimic gestures in the lab, what does this mean for the home environment? What does it mean for early learning? So we actually asked them, and once we asked them, we also compared their judgments to what we were with the labels we were getting, right? That research definition of, is this an imitator or non-imitator session? And so here we did this in 2 different ways, because some people contributed a lot of different sessions. We either looked at only the first included sessions, or only those 3 people who contributed a lot, so 9 or more, just to have kind of, to avoid biasing the analysis either way, to, people contributed a lot of sessions, or not taking advantage of that rich data set we get from people who did participate multiple times. Now, did the researcher definition of imitator versus non-imitation session agree with the caregiver perception? The answer was no, not at all, in fact. And so that was really, really surprising to us. So here what you're seeing is the caregiver judgments on the horizontal axis, and then a number of sessions that kind of were at each of those confidence judgment. These mimicry judgments. So either high confidence child wasn't imitating, or too high confidence child was imitating, from left to right. And then red is the session is labeled as mimicry based on that researcher definition. Green, it's not. And you can see this just doesn't align at all. And that's also the case if we look at people who participated multiple times. So that was really, really surprising to us. And so here we kind of shifted to exploratory mode and just looking at, okay, what's going on, and doing more exploratory analysis. And so here, what this looks like is that it actually looks like what's going on. It's not that there is no information, no data that caregivers are picking up on. It's just that those 2 measures appear to be biased in opposite direction. So in the slide right before, right, you saw the percentage of sessions that were labeled as mimicry or not, and mouth opening sessions tended to be labeled as imitator more often, perhaps because newborns just make more mouth opening. But caregivers actually were biased in the opposite direction. So if they were randomized as tongue protrusion modelers, they were actually more likely to say that their newborns was imitating them, which I'm not sure what to make of this, but and again, this is a small sample, but this is what we did find. And that was the case in kind of just looking at the first session for everyone, and also for 3 people, but then there's just 1 tongue protrusion imitator. Modeler, sorry. And for those 3 people who did participate a lot, we also had this opportunity to look at session by session, the same person. Was there any connection between the newborn behavior and their judgments? And so here the answer seems to be yes, but again, we're even smaller sample than before, which is literally 3 people. And in the 3 people, we did see either a significant or marginal association between the number of, the rate of matching gestures that the newborns were making. So say more tongue protrusions if the caregiver was modeling tongue protrusion, and how confident people were that their child was mimicking them. So it's not that caregivers aren't picking up on anything, it's just they appear to be biased in different ways from our researcher definition, just perhaps something to bear in mind. All right, so conclusions. Essentially, we found that it was feasible to conduct this sort of study online with newborns, which wasn't totally a given, given how hard it is to study newborns. There are lots of opportunities for improvement. So I think the question of automated video processing would be really, really important for scalability, particularly if we're talking about collecting hundreds of videos, and particularly for small labs with not necessarily a lot of trained students and staff. That for us, at least, that was one of the biggest bottleneck, I would say. Repeat participation, not sure if you're considering this, but if you are, one lesson that we learned, and I think I've heard from other people doing online studies, it's not an uncommon one, is that if we let people to just choose whether or not to repeat participation, some will not do this and some will do it 20 times. And so having some sort of target may help here. Again, this is another, probably shouldn't have reinvented the wheel there, but this is what happened. And if we are going to involve caregivers again, or even across labs, right, having that standard gesture frequency would be important, as would be kind of making sure that we're not underrepresenting caregivers without a bachelor professional degree. For example, because our sample was, because it can be important for interpretation too, right? Because the sample was so educated, who knows if the reason they were more likely to think their newborns was mimicking when they were doing tongue protrusions because they've read your papers and they know. and so they have more expectations around that. Right. Okay, so we did not find evidence of group level mimicry, which doesn't say anything about individual behaviors. It doesn't say anything about whether they could do this in the lab with an experimenter and so on, but this is what we found for this paradigm. All right, I won't cover that. All right, so and I think the one thing that was quite surprising to us, but may have some implications about what measures we want to include in this project is, what do we mean by like what type of mimicry matters, and kind of how do we define mimicry, which is and in particular, is this something that is aligned with the sort of thing that caregivers actually pick up on, and if not, then do we want to collect different measures and kind of see what's the best way to capture what we want to capture. It's just again, very naive comment because I'm not a neonatal mimicry researcher, but all right, so that's what we learned. So hopefully there's something here relevant to this call, but I think for me, the points that are relevant to this call, since there was some mention of doing online studies, is probably it would be more relevant for this group to have things like video, standardized video stimuli doing instead of caregiver movements, right? But we did find that it was relatively feasible to do this. Obviously, there would be more questions about things like if we want to have people using infant boosters, an infant car seat, for example, standardizing this sort of things. Not sure how that would work online, but at least the things that I would say that the things that we were worried about, things like, will caregivers be okay putting their webcams in the right angle? That wasn't too much of a problem. But we ended up having problems that were unanticipated, things like caregivers holding movements and so on. So with that, I'll stop talking. Thank you for listening. And if you have any questions, comments, or feedback, really happy to hear this. This is very early finding. Some videos still haven't been, video coded. So this is very, very rough, and feedback is appreciated.
Okay, great. Thanks, Laurie. Awesome. Does anybody have any quick questions or anything before we move on to our last presenter? Yeah, Nazim, go ahead. Oh.
Hello. Yeah, thank you for the nice presentation. It's not a question, it's actually a comment. I mean, I find your finding that, parents actually see imitation, even if there is no imitation, very interesting, because in this literature, one of the reasons given for this neonatal imitation capacity is that it helps social bonding, but maybe you don't need an imitation capacity to get this social bonding going on. The parents are already biased, so even if there is no capacity, they'll overinterpret their kids' behavior. So I thought that was very interesting.
Yeah, that was exactly our motivation. If this is important for bonding, then we want to see if caregivers pick up on this, right? Now, here we can't, I mean, again, like, I'm not sure we have a good ground truth here because of the baseline rate issue of our measurements. So just bearing that in mind, right? Just because the session was labeled as mimicry or not mimicry by this definition doesn't mean that this is the ground truth and parents are wrong. But I do agree though that it's, at least if we're worried about consequences for bonding interaction, then having some sense of perceptions by caregivers would be important.
Yeah, and also it was very interesting that caregivers also got stuck with tongue protrusion, like Kathleen and I did. It's like,
yeah. Although again, it's maybe education.
Is that's the weird one. Okay.
Yeah, that was really unsurprising. And again, small sample, I may not replicate at all, right? But this was very surprising to us. And one thing that I forgot to mention, because there is this whole question of what are we measuring, how do we agree what counts, and so on. I think the proposal to have videos on data would be really important here. Because then different labs can use different coding schemes and see if they have different conclusions. And in fact, for this project, we're also planning to have started putting some videos on Databerry, again, with easier since we are based in the US, right? So we don't have to handle GDPR. But I think that's also kind of an important part of the project.
Okay, great. I think we'll move on to our final presenter for today, John Redshaw. So I'll hand things off. Go ahead, John.
Okay, so can everyone see my my screen there? My presentation? Yep, excellent. Okay, so firstly, I'd just like to say thank you to Heidi, Mike, Sumeet, Virginia, and Mark, and everyone for coming together in the spirit of collaboration here. I think it's a really great thing to do. It's a wonderful initiative. So what I'm going to do is I'm just going to run through the most recent paper to come from the UQ Imitation team. So this was our meta-analysis that we recently published in Perspectives on Psychological Science. So yep, like the others, just a 10-minute talk. I'll provide some brief background. I'll give a summary of the methods and the findings. And I'll conclude with what we think this meta-analysis might mean for MB6. Okay, so yes, so the authors on this paper were the core UQ neonatal imitation team. So everyone here has made critical contributions to the various projects and papers along the way. This one was led by our resident statistical guru, Jack Davis, who was at Cambridge at the time, and Virginia Slaughter was the senior and corresponding author. Okay, so I assume most people will be familiar with our original neonatal imitation project, but just a quick recap for how we became motivated to conduct this meta-analysis in the first place. So the original study was funded by the Australian Research Council, a grant to Virginia, Mark, and Thomas, and it wasn't designed to test the existence of neonatal imitation per se. So it was actually assuming the existence of neonatal imitation, and the idea would be that we would track individual differences in neonatal imitation and see if this could predict later social and cognitive development. So we tested 106 newborns at 4 different time points. 64 of those newborns contributed to all of those time points. We looked for the most common gestures reported in the literature. So these were 2 facial gestures, tongue protrusion, mouth opening. We had some facial expressions, happy, sad. We had a couple of manual gestures, 3 vocal gestures, and we also included 2 non-human control models. So tube protrusion and box opening matched to tongue protrusion and mouth opening. So after multiple years collecting and coding this data, we then spent even more time double checking our coding, screening for infant attention, fidelity of the model, looking for order effects, all of these types of things. And we just had to come to the conclusion that there really wasn't an effect to be found. So what you can see here is the 9 actions as coded from the 9 baby responses. So where it says tongue protrusion, that's referring to the baby's tongue protrusions. And what we have in black are their actions in response to the control models. So there's 10 black lines for each of the actions. And the red line is the response to the matching model. And across these actions, what you can see largely is that the red lines are pretty much smack bang in the middle of the black lines. We're not seeing any greater responses to the matching models than the non-matching models. Now it has been pointed out that the tongue protrusion one is a special case. So if you compare the red line to the average of those black lines, the red line is higher. But when you compare the red line for tongue protrusion to the black lines for mouth opening or happy responses or sad responses, you don't see a difference there. So there didn't seem to be a particular thing about tongue protrusion towards the face either. But of course, because neonatal imitation was not the question, but an assumption, there were some reasonable questions raised about whether the method we use was appropriate to find mere existence of the effect. Because we were assuming it existed, we might not have used the best method to detect mere existence of the phenomenon. So what we set out to do was we did a meta-analysis looking at data reported in 26 papers, 33 different samples. We had 336 effect sizes. This meta-analysis included data from over 1,000 babies and 16 different labs. And so here's the upshot, just to get straight to the results. You can see the forest plot of all of these neonatal imitation studies that we included here. Now what we have here around is the black line represents the null effect. So that's what you would expect if there is no such thing as neonatal imitation. And the red line is the average effect of all of the different studies, all of the different samples that we included. And when you look at that red line, that represents a significant and medium-large positive effect. So the D is between 0.5 and 0.8, so the traditional medium and large effect sizes. So taken by itself, that would suggest that neonatal imitation does indeed exist. But of course, you have to also pay attention to the variation. So what seems to be present here in this forest plot that you can see is there's a large group of studies that are clustered around that null effect line, but there are also a large proportion of studies that find large positive effects. So obviously the next question to ask, well, is what might it be about these studies that are finding large positive effects? Is it something about their methods that enabled them to detect imitation effects when the Oostenbrück study and these other ones did not, because they didn't have the same methods? So what we did was we looked at the literature and we looked at claims about factors that might influence the size of the neonatal imitation effect. And there are a lot of claims out there. And because there were so many and we didn't want to have false positives, what we decided to do was to just include the variables that were identified by at least 2 sources within the published literature as being factors that could influence results. So I think there were 5 different papers we looked at. And among those 5 papers, anything that was mentioned twice, we included in the meta-analysis. So we included all different types of variables, so aspects of the gestures presented to the infants, aspects of the response time, the total number of models in the study, total experiment time, so what the comparison condition was, experiment setting, identity, and so on. We included many different things. And the upshot is that none of these methodological variations had a significant impact on the overall neonatal imitation effect. Yes, so we took quite a liberal statistical approach here. We didn't correct any p-values. We didn't adjust the alpha threshold or anything like that. And even taking that liberal approach, none of these moderators crossed the 0.05 threshold. The closest one, which was 0.62, I think, was whether the infant was in a seat. But again, that one was not a significant predictor either. So post hoc, so we did not pre-register this. We also looked at whether researcher allegiance was associated with the effect, and it was. So it is certainly the case that this effect has been replicated by several labs. So six out of the 16 labs reliably do find this effect, but 10 out of 16 did not find significant evidence of the phenomenon. Another thing I'd like to draw people's attention to is the large confidence intervals here. So even if you look at UQ, with all of those babies that we tested at all of those different time points, within our 95% confidence interval there, we still have a 0.5 effect, right? So I think that's what ManyBabies will be fantastic for, is for narrowing these confidence intervals, which are just par for the course in infant research. We know infant research is very noisy. We know there's wide confidence intervals, and I think ManyBabies is going to be very useful for that. So conclusions from the meta-analysis. We did find a positive and medium-large overall result for neonatal imitation, but there's substantial heterogeneity in the literature, and that heterogeneity cannot be explained by captured methodological variation. So I've underlined captured there because it's very important to point out. So meta-analysis is obviously a very crude method. We have these dynamic processes, and we're just coding that from method sections, right, and turning those dynamic processes into ones and zeros. And when we do that, we did not find any evidence that those methodological variations were associated with the effect. So how can the meta-analysis inform MB6? So in and of itself, it can't. So we didn't find any evidence that these methodological factors as coded from method sections do explain significant variance in the imitation effect. Therefore, there's no methodological factor that we can point to and say that this is what MB6 should be doing. But of course, what I think what we would want to be doing is asking people who have found positive effects and seeing if they can help us kind of identify what they might have been doing in their studies that did lead to these positive effects. But of course, setting aside the meta-analysis, the design, of course, should still follow these established principles that were originally identified in the Meltzoff and Moore paper, and many others have pointed this out. So we want a cross-target design with at least 2 target actions. Again, I think for something like ManyBabies, it might be ideal to go with exactly 2 target actions just for feasibility purposes. Tongue protrusion and mouth opening, I think, the traditional ones sound like a good initial starting plan to me. And of course, in order for it to count as compelling evidence for imitation, you do need to see this cross-target effect. So in order to rule out explanations like those that have been raised by Susan Jones, you want to see that action A in response to model A is greater than action A in response to model B, but also, critically also, the rates of action B in response to model B are significantly greater than the rates of action B in response to model A. Now, how you precisely operationalize that DV is up for question. So I think, as in the previous talk, we could look at mere frequency of the actions during the testing period, or we could look at the increase from baseline. But no matter how you operationalize it, I think you need to find these cross-target effects in order for it to count as compelling evidence for imitation. Yeah, so I'll stop there. That's my talk. And if anyone has any specific questions, feel free to jump in.
All right, great. Thank you, John. And that concludes our presentations for today. So I will open it up to all of our panelists. Now we can just have some conversations. You can feel free to pose questions to each other or comment on things that you found particularly interesting or compelling. And I'd also like to invite those of you in the audience. You can either put a question in the Q&A function or raise your hand, and I'll be able to give you the ability to ask your question directly to the panelists. So with that, I'll step aside and let you all talk to each other. Thanks again, everyone. Those fantastic presentations. Yeah, Virginia, go ahead.
Thank you. That's a great start. I want to react to Andy's suggestion that we go with 6-week-olds. I do appreciate that they're a lot easier to manage, but I think if we go with babies at that age, we won't get the kind of answer that we want, because ultimately what we want to know is the capacity to imitate there from a very early age? Is it inborn? And if you look at the alternative model, say Cecilia Hayes's associative sequence learning model, that would predict that by 6 weeks, babies might have had enough experience of producing gestures and seeing those gestures mirrored to them to have created the neurological basis for imitation. So that's what I wanted to say about that.
It doesn't seem that in the study that Jonathan just reported that you supported the Hayes hypothesis.
So yes, we in the meta-analysis, I'll just clarify, we included only infants aged up to 6 weeks. Again, I would have to check the supplementary materials to be 100% sure about this, but I do believe it's correct that we did not find an age effect between 0 and 6 weeks. But having said that, I can see Virginia's point that yes, if we did find an effect, I'm not sure that would be convincing to some of the more, hardened skeptics about the phenomenon that it's innate if we found an effect at 6 weeks. But I also appreciate Andy's point that, well, if we found an effect at 6 weeks, we could then go back to earlier and see from there.
Mike, go ahead. Yeah, and please don't wait for me to call on people. Feel free to jump in.
Thanks. So let me just mention that I'm hearing a couple of trade-offs articulated here that have come up in a bunch of other ManyBabies projects. I don't know the right answer, but I can tell you a little bit about navigating these trade-offs. I mean, roughly the big trade-off is between variation and standardization. And so standardization of methodological decisions, it increases comparability of results across labs. So age could be one example of that, although it's certainly a theoretically more loaded one. So that's a positive. And if you know the right answer about a particular methodological decision or population sampling decision, then you increase your yielded effect size, right? So if you know that 6 weeks old, yield a larger effect size, measuring only 6 week olds is going to get you a bigger effect overall. Similarly, if you're sure that car seats are the right, and again, I don't have an opinion on these particular decisions. I'm just using them as examples. If you know that car seats are are better, then you get a bigger effect by using only car seats. Maybe this is obvious, but the alternative is you allow these things to vary and you know that they matter, or you think that they matter, but you're not sure about the right answer. The positive of that is that you can then plan for hypothesis tests around these methodological decisions, following the approach in the meta-analysis, and you can use them as moderators. So for methodological decisions like the car seat, you get to ask, is there a difference between car seat and no car seat? And for age, you get, if you, as Krista Byers-Heinlein suggested in the comments, if you allow age to vary continuously, you can use that as an analytic variable in your models. So there are some positives and to allowing for variation, but you get a kind of limited statistical budget, I would say, for these. You can't put in 1,000 moderators and expect to actually understand them. In fact, you could average out, if you put in a lot of bad decisions, you could average out some signal very easily in that case. So you have to spend your statistical power sort of wisely in this case on decisions that yield insights about future methods going forward or yield theoretically relevant variation that you want to bring forward to, sort of help you understand the theoretical underpinnings. So I don't know at all on the methodological side which of these are the kind of really key ones you'd want to fix versus which you'd want to allow to vary. The last concern, though, is that there may be certain methodological decisions that lead you to decrease the amount of labs or participants per lab that you get. And so age is one of those. The broader you sample, probably the easier it is for labs to recruit. And so on the, I think probably labs, if you ship them a car seat or give them funds or even just tell them they have to buy a car seat, they'll buy a car seat. And so maybe if that's the right decision, you just do that. On the other hand, with age, you might actually yield a larger sample, which actually counteracts the negative effects of having a weaker signal in some younger kids. If, labs were going to give you the same number of 6-week-olds, but they're also now going to give you 0 to 5-week-olds as well, maybe that's worth it in that case. So some of these questions are best answered by simulation, some by discussion, and so forth. I'm just pointing out that there's this kind of range of trade-offs that we've had to deal with in a number of these projects where we, in some cases, we've erred too far on the side of allowing variation. In other cases, we've kind of made a particular choice betting that we knew the right answer. And there's no one right solution here, but we can sort of bring evidence to bear on navigating them in some cases.
Yeah, I think I would agree with that point on the practical benefit, right, of having a broader range. And it seems also just from listening to the great discussions here that there would also be a theoretical benefit, right? Because it looks like different theories make different predictions. No age effect, a decline, an increase, right? And so we would get to find out. Now the one question, I guess, from again naive perspective with regards to ManyBabies is pooling data across labs, because if each lab measures this thing, sampling a kind of a broad age, and if we do decide to, if this group does decide to allow some sort of age range, then the pooling across labs, I'm not sure how that works if infants come from different ages, like looking at an age effect. From like a statistical perspective, if you do get to include each individual infant as an observation, it works pretty easily. But I haven't read enough ManyBabies papers to know if that's usually how it works.
Yeah. I think I'd just like to concur with Laurie there. I think, from my perspective, I think the best approach would be to let age vary, perhaps between birth and 6 weeks, because I can imagine the logistical issues for labs trying to target, 60 infants within 3 days of 2 weeks and 60 infants within 3 days of 6 weeks. That might present logistical issues which would harm data collection when we could get twice the amount of data, allow age to vary. And if age is not having much of an effect at all, if any, then that can be captured with a moderator analysis. So I think that would be my hunch, but of course I'm willing to hear other perspectives on that.
Yeah, but this seems to be, I think there's 2 issues. One is about standardization, advantages of standardization versus variation, and there's trade-offs. And another one is kind of theory and related to why we're here trying to do this study within ManyBabies. And so maybe start with so not trying to drive anything to conclusion, just airing things. If you take across these studies, what has been reported in Australia, generally, this group, is that there isn't any systematic stimulus control such that if an adult sticks out their tongue, the baby sticks out their tongue at a higher rate to that than they do to other controls. That's what's being written. And that's true, says the group, at 1 week, 2 weeks, 6 weeks, 9 weeks. It isn't found in the early period. And another group has done autodigestive stereotypies and says that the stereotypies, that mechanism pertains up until 4 months or some period of time. So it's not that it's changing at 6 weeks. We're not gonna, if we show something at 6 weeks, it's not what's being predicted by that theory. And on the home study, I think the home study and the photography, the videotape home cams are, you know, that's really brilliant, and it could be used quite profitably that we can talk about in future meetings. But in any case, so I think that that's kind of useful, but the data that's been reported, at least when scored by certain criteria, is showing no effect. And so people aren't finding that if you stick out your tongue, babies are sticking out their tongue, or at least not doing it differentially more to tongue protrusion than a bunch of other controls. And people are saying that about 6-week-olds, including 6-week-olds, also 2-week-olds, but also 9-week-olds. And so one question is, what would happen if the group got together and did sensitive eliciting conditions, and all of a sudden found that tongue protrusion is elicited more to tongue protrusion than to a variety of other actions? That's got to be informative to this group. Now, it would also be informative to you. It's important, as John pointed out, to show mouth opening peaks and mouth opening various other controls. But right now, the report is the behavior isn't showing more tongue protrusion to tongue protrusion than other gesture. So one possibility of how to proceed there is just to see if people can find that and get used and have techniques that elicit that. It's under stimulus control. Sticking out your tongue makes it go up versus poking out a pen, which has been used as a control, versus face models, which have been used as a control. And when you have that, then find out at 6 weeks, then find out other things. That's the root of the suggestion. If people could demonstrate that, that would be a big change of the literature right now. Then that group who can find it at 6 weeks should go down to 2 weeks, and then they should go down to newborns, newborn being hospitals. Not hospitals, it could be at home, but anyway, babies in the first 3 days of life. So that's the only point about that. So the standardization and variation is that, yes, you could have a lot of data points, but if you could put all your firepower on finding, you know, all the degrees of freedom and all the N that you can get, where on a critical question, that's an interesting first start. So I apologize for saying that too strongly. It's just a bid. I know it needs to, people don't necessarily buy into that. I get it. But I'm not sure the absolute critical question for this group is, do newborns imitate? Because people are saying 6-week-olds don't imitate. I mean, it's just, that's the claim. So boy, if 6-week-olds imitate, and all we're arguing about, 6-week-olds do it systematically, sure, but 4-week-olds or 2-week-olds don't. That's a very different debate. Now, it is true, I am predicting newborns imitate, absolutely, not backing away from that. That's true. But there's big news here. If everybody just says, well, 6-week-olds, no problem there, then we knew they would do it.
I just wonder though, like what this project is targeted at, right? Surely is the question of whether this is an innate phenomenon. And I think, to go back to Virginia's point, if it's 6-week-olds, and say we do find a positive effect, there will be, it's not going to convince the most ardent skeptics. And given that the theoretical question we're interested in here of whether it's innate, I do think it is a very interesting question whether the young ones do it. It's not just, well, 6-week-olds do it. That changes people's minds. I don't think that's still the critical question that this particular project is targeted at.
Just a quick response to that, quicker than my last. This has come up in other projects as well. There's sort of a 2 steps forward, 1 step back type of thing that happens. So in ManyBabies 2 on theory of mind and infancy, there was not consensus that the a negative result would be kind of probative for the theory unless we had a prior positive result. So the first registered report that is actually now being carried out is simply about belief tracking, not about false belief. So the idea is to establish that we simply have under clear experimental control belief tracking, and we can get action anticipatory looks based on that. And then if that's successful, we will then go and try to test positive or negative false belief under various circumstances. So in that sense, doing a 6-week study, if it were believed that that was a more feasible study to run, could be, as Andy mentioned, really just getting the measurement right, making sure that you can have control over this phenomenon. But it's hard. It's very hard to say. Kind of depends. Also, do you need 1,000 babies to do that? If so, maybe it's not a great study 1 because study 2 might be a long time in coming. Do you need 100 babies to do that? Maybe that's more doable. Especially if there are a number of participating labs.
I like the idea of letting age vary and letting the labs make use of the participants that they have access to. So, we can actually get a lot of very young babies because we regularly recruit through Pregnancy and Babies Expo. And so we get people signing up to participate when they're still pregnant. So we can get a hold of quite young babies. I also think that Andy's suggestion of doing a pilot study, videotaping everything, and then sharing that around before we move forward with the real study is crucial.
Just to switch the topic just a little bit here, do we have a sense of how many groups might be interested in participating in this type of study? So in thinking about previous ManyBabies studies, when you test older infants and children, there are a lot of groups out there studying older infants and children, whereas newborns and neonatal imitation in particular were kind of niche. So I have some concerns and some doubts about, are we really going to be able to convince people to recruit this particularly difficult population to recruit and to test this particularly difficult population to test? And are there things we can do, like incentivize it? I know here we have to pay very generously to these families of newborns to get them to come in for every single visit. Are we going to need some grant funding to kind of back us up here and even making this study feasible on a larger scale and reimbursing these families? I don't know if anybody has any kind of sense of that yet, but that was kind of a question I've been thinking about.
Hi there. So I just wanted to chime in there. I think you have a great point. I think it's a lot more difficult to recruit newborns than it is older infants. But I think that based on our experiences with the Pregnancy, Babies, and Children's Expo, we could provide some suggestions on how to best approach that and how to maximize recruitment for a specific age group, whether that's 2 weeks or 3 weeks. A lot of the previous work has had pretty low sample sizes, and that's also because it's quite difficult to get moms to agree with bringing their babies into us when they're so young, which is also why I think a lot of work has traditionally done home visits rather than have mothers bring their infants into the lab, just because they're at a very critical time and they were just born, and some moms might be a little bit afraid of bringing their children out to labs and things like that. So I think we would have to have a conversation with the interested labs to see what their thoughts are, and if they have any local events where they could go and do recruitment of expecting mothers like we do here in Australia, and definitely provide some guidance on that. So just to give you some perspective, we did a recruitment event over a weekend, and we ended up getting 150 expecting mothers and families to sign up for it. And that doesn't mean that all of them will be interested in participating. And that's something that we have to figure out as we email them and we get in touch with them closer to their delivery dates. But it was quite a successful event, and we've done that twice now. So we'd be happy to share any of the guidance and support for other labs who might be interested in getting newborns into their labs for particular studies, and we can share our insight on that as well. Happy to do it.
Yeah, I mean, just coming from a group that's been doing this for 7 years now, we've gone to lots of baby expos over the years too, and it's not always easy to get newborns to come in. So maybe at your site, you guys have a lot of luck, but not all sites are. It's not always so feasible. So just kind of anticipating that that's going to be a big hurdle, and this could be a 20-year study. If that's what you want to venture into, that's fine. But this could be really a long-term kind of commitment that we're putting time in. This isn't going to be a fast 1 or 2 year turnaround study.
100%, completely agree. Yeah.
Yeah, and from the online perspective, again, here we're really not set up to test newborns imitation in the lab. But when it comes to online recruitment, you can recruit more. It's more scalable, but it's not free. We were compensating more than is typical for Lookit, and we did recruit using paid ads, which worked. But again, we're not free. And so if we're talking recruiting, 100 newborns per lab, then that's going to need a budget that not every lab. I know we wouldn't have the discretionary funds to do that. So I think finding either kind of allowing smaller samples from different labs, including the online environment, or planning to obtain funding would be important, because particularly if we're talking smaller number of labs, and it's perhaps usual for ManyBabies.
So just to kind of add a couple pieces of context on the recruitment issues that we've described. The first is that we've found, kind of from a statistical perspective and also from a kind of an inclusiveness perspective, that if we put out a call for labs that doesn't have a minimum sample size, we can often get labs that are willing to contribute, say, 10 babies. And actually, if we're interested in lab-to-lab variation and estimating and trying to control for that, that actually is very helpful to get different labs, even if we don't have a ton of babies from each lab. So the highest level sampling unit, which in our case is the lab, actually, the N at that level helps you estimate the variance of lab to lab differences. The second thing I want to mention is that it is in principle possible to do a sequential testing design, especially if you're thinking about a study 1, study 2, where study 1 is get the thing under experimental control. So it'd be possible to register a sequential design in which we check every time we have a kind of upload of babies or something, and compute a running Bayes factor for whether we see evidence for the phenomenon being under experimental control. That might be warranted here because of the costly nature of the study and the desire to move to study 2 if there's a positive finding. We haven't done that in ManyBabies, but there's a number of nice studies coming out now that have, there's a beautiful paper by Nivi Mani in which she does that for 3 phenomena and kind of very quickly and efficiently replicates or fails to replicate those findings. Finally, on the funding note, I think it would be great to pursue funding here. And we'd love to work with the leadership team to try to do that. We've found that national awards, NSF, ARC, DGS, whatever your national funding agency, that those are very good for hiring staff. And national funding agencies are very bad at giving small amounts of money to many institutions to let them do their work, unfortunately. So my suggestion would be that we, as we assemble this project and the leadership team, we brainstorm about staffing expenses and whether those could be funded under proposals to national funding agencies, and recruitment expenses, which could be funded by foundations. So for example, ManyBabies 1 received a $50,000 grant from the Association for Psychological Science, re-grant from another foundation. And that was sufficient to give, I forget, several dozen $2,000 awards, or maybe sort of max of $2,000 or $3,000. And for a university, it's just annoying to, like, they can't cut that many checks and send them out. But if you get a foundation, they'll often be willing to work with you and send enough money to pay for those Lookit studies or those newborn recruitment fairs or whatever. And that's really what we're looking for. So we could brainstorm about how to do that, but that would be one way to get, kind of moderate amounts of compensation to incentivize labs.
Yeah, and I just want to quickly sort of add to what Mike said and just say for our other projects that are going on, the typical ask from labs is like 16 to 32 babies. So we haven't had any, some labs contribute more than that, but we haven't had it be a standard where the ask is higher than that. And that also helps make the barrier of entry a little lower. I can also imagine, trying to frame it, the recruitment sort of added burden, try and sell it to people the other way of like, this will help you fill out your subject population as the babies get older, and establish contacts with parents early on. I know in my own experience, it's always easier to get people to come back into the lab than it is to get them there in the first place. And so, yeah, maybe there's some creative thinking there too about ways to expand subject populations. And if you guys are able to offer some of your tips and expertise in that field as well, I could imagine that being helpful to labs that aren't used to testing such a young population.
Just to jump in again, I think all of these logistical issues that people are pointing to here, they're only amplified if we restrict age. And I feel like it's such a small loss to let age vary, and it's something that we can actually measure statistically, empirically, for such a large gain that we can increase our sample and give ourselves greater power of detecting imitation effects at all. That's just my hunch is that all of these logistical issues that are being raised are really great points. And I do think that one simple solution would be to let age vary if everyone is on board with that.
I'd like to bring up something different, which is the stimulus, the model stimulus that we use. In the meta-analysis, there were only 2 studies that used video presentation, but even still, with those 2 studies, there was a significant positive effect for video presentation. And when I talk to non-psychologists about this work, which of course everybody is interested in, oftentimes people from other sciences say, how can you possibly do a controlled experiment where you're interacting with babies? I mean, obviously the baby is going to change the way you interact, which is, of course, an excellent point. As much as we try to be neutral in our model presentations, all kinds of things can creep into that interaction. So I'm wondering what people think about the idea of standardizing the model presentation by having video.
Well, in imitation, there's a famous phenomenon, as you know, Virginia, called the video deficit effect in imitation. And so there's a big literature that infants, they are at older ages, but infants are significantly poorer imitating from a flat screen, from a television, than they are live. It's not my lab. I mean, we've done labs. We've done imitation from television in my lab in older kids that's successful. But in any case, many labs have done this and in older kids and found deficits. So again, it's a sequence of studies. It's a question of if one wants to put the firepower on finding out whether there is this phenomenon, this potentially social phenomenon in live 3-dimensional world, one would try to find that out. And then one would say, as in any study in developmental psychology, not all studies are on television. I mean, people who's doing object permanence in person and done attachment studies in person and so forth. So what you would do is see if you could find it in a live naturalistic type of thing under experimental design, and then say, film exactly what you did live that worked live, and now put it on a two-dimensional screen and see if you get it. If you do, that's terrific because you can manipulate a lot of things. Once it's on television, you can manipulate everything. If you have it live and it falls to chance on two-dimensional screen, which is what the video deficit of imitation did, then you'd say that babies maybe social responses responding to somebody else, to a person, is different than to a representation of a person, to a flat screen, as Pat Cole found in speech perception and learning and, you know, phonetic perception. She did it live and videotaped what was happening live, and the phenomenon fell to chance when it was done on a screen. So that's news too, and that's terrific. I mean, that's a very interesting thing in speech perception. You can't get the same thing from a screen. The audio track's exactly the same. Audio track's identical. Babies respond differently to television. So I do think it's extremely valuable to do things from television. I have done it from television. It's difficult to see that that's the first move on the hardest question with young kids when there's published literature about video deficits. I think you want to put your firepower on seeing if you can find the phenomenon across many labs with a standardized procedure that people don't think is artifactual. Like if it came out here, we would accept a finding. You know, that would be a big step forward.
Could I jump in? I was just going to say that, sorry, can you hear me?
Yes.
Thank you. Okay. I was just going to say that the age is really going to matter, whether the two-dimensional screen is going to differ from the live performance, because certainly at birth, you don't have binocular vision going, right? And so it is, I think, really unclear whether early on the two-dimensional screen is in fact going to be different for the infants than the three-dimensional real live person. So I think it depends on age to a certain extent, you know, whether you can switch these things out. And I'm not sure that it has been shown that for early on that there is a difference. Certainly there is a difference later on when the, you know, infant's visual capabilities, you know, zoom. So yeah, age really matters for that one.
Yeah, I think I agree with Kathleen there. It seems like we want to make sure that the babies are interpreting the model as a model in order to make sure that we're eliciting, you know, we're giving them the best opportunity to demonstrate the phenomenon, I think. I had a different question. It was about Andy's suggestion that we use a between groups design. And from my perspective, maybe I'm missing something here, but I wonder, I listened to the point about carryover effects, and I appreciate that point. But I do wonder if we can have our cake and eat it too. So say we were to use a within groups phenomena, a within groups design. So that way we're using exactly the same participants for, you know, across actions. If there are carryover effects, well, we can still just look at their first action, right? We can treat it as a purely between subjects study. So my hunch would be to use a within subjects design simply because we can look at carryover effects. And if they're there, we can, you know, describe the nature of those effects with a large sample. And if Andy's problem is a genuine concern, then we can just look at the first action that was shown to babies and treat it as a between group study.
Jonathan, I appreciate that. You're exactly right. You need counterbalance order, and that's what we did in our studies. Then you can take first trial and you can analyze that just as you're saying. That's very good logic, and one can get at it. And so there are some values in doing repeated measures, which we should continue talking about, which is you have that same baby in that same state and you test them twice and you know, in a row, and you can make that comparison. So I'm with you about that. And so we just need to talk about it more. The independent groups one came along with it that are strongest effects on imitation, because we modified, we changed our study from when we did newborns to when we did 6-week-olds. We got a stronger design when we were able to see the same subject over repeated days to get a more stable measure of that child, not just what they were like at 11 o'clock on the morning on Tuesday when the mother first came in. So we got 3 assessments of the kid. And by doing that, we were able to get a fairly long test period, testing data, a lot of data on that kid on a gesture. And we found that having that more of those data on an individual child was useful. So it's a trade-off, not only answered by saying you take the first trial, because if you do a repeated measures thing, your length of study is only so long and the first trial is half of that. When we did one segment over 3 days, we got an enormous amount of data on one child because they didn't habituate. It wasn't in one session. They didn't fall asleep. They were in a terrific state each of 3 days. So it's partly that. I do think the discussion should continue about it and is not decided. I'm just trying to get things on the table that we saw in our own lab, because the goal of ManyBabies must be here to try to have the best eliciting conditions we can have that wouldn't artifactually produce a positive result. You want to give, that's what we're trying to do, to see under those best conditions, can we get the babies under experimental control in the way that's consistent with imitation or not? So I think you were right in what you described, Jonathan. I'm just amplifying with some other things.
No, understood. Thank you very much.
Yeah, I just had a quick question for John and Andrew. So I completely understand looking at it from a within subjects design. And I just think logistically it might be a bit difficult if, like, and this is just a general question, if parents come in for the first 2 sessions, or we go in for the first 2 sessions, but on the 3rd they're not available, or the infant becomes distressed, we kind of lose out on that data. We're more sensitive, I guess, to attrition whenever we look at anything more than 1 session repeatedly. And also, I think that issue of that Andrew brought up, which was great, that, you know, not telling parents that we're specifically looking for imitation or looking at imitation is important, because of course we might see that some parents are practicing gestures in between these visits. How would we be able to control that if, let's say, we went into the home 3 days successively? It's just a general question. I'm just curious to see how we might be able to, you know, add some control in there for those sort of things.
Sorry, was your question if you go into the home 3 times, or?
If you have, like you mentioned, if you go in 3 days successively, so they're in the same state, but across different days.
Yeah, that's a great question, Sumeet. I think you're thinking about the very good problem there. What we did to solve that in the lab is we did not inform the mothers that the test was on imitation, and the mothers did not see the gestures being shown to the child. So they were blind to what the gestures were.
Okay, that makes sense. Okay, I was just wondering how you.
They couldn't have affected it at home.
That makes a lot more sense. I thought they were perhaps sitting behind the infant seat where they could see the gestures of the experimenter. Okay, that makes a lot more sense. Okay, so that'd be something that we could definitely include if we control it that way. Cool. Thank you for letting me know. Appreciate it.
Yeah, so I think this is an example of something that probably could not be implemented on Lookit. I would have to go back to the kind of guidelines again, and maybe this is a discussion that could be had with the kind of persons in charge of Lookit. But as far as I understand it, they don't allow with the duplicity. You need to disclose what you're looking at, even if in general terms. And certainly we would want to show them what we're showing their child. So maybe there would be a way to say, your child will see one of those two videos, right? And it's a secret which one, but at the very least, we would need to, I think, disclose that we're interested in babies' reactions, to put in the most general terms possible, at the very, very least. And so I don't think, I think the approach would need to be, if we're doing an online component, we wouldn't be able to, parents be completely blind to this.
We've called it the funny faces study. And actually we've gotten quite a few newborns that smile and laugh at us. We make funny faces at them. So the parents just think it's, their infant's positive reactions to somebody making funny faces, right? That could be a good way to kind of hide what your specific predictions are. Now, it doesn't mean they're not going to go home and make those funny faces at their own baby, right? Of course, but.
Yeah, that's a tricky bit. And that's kind of why I thought about just doing like a cross-sectional design where you just go in once, parent really has no idea or any opportunity to have any guesswork involved, and just getting that data point and then moving on to a different participant. So that was just always a concern. It's a concern in adult literature as well, whenever you're looking at behavioral mimicry between social interactions or automatic imitation. So obviously it would apply in this case as well. So yeah, so that's interesting. So funny faces. Are there any other kind of tools that you guys have told participants that have worked in the past as to what we're looking at in the lab?
I mean, I was thinking if they do a different condition each time they come in, right? So it doesn't matter if they've been practicing what gesture they saw when they came for the first visit. If when the baby's coming in for the second visit, they're seeing a totally different gesture. So that would kind of solve that issue across test sessions, across days. They saw the different conditions.
Right. Okay, makes sense. Yeah, and that would still, we would still run the same issue on Lookit. We would have to disclose the specific gestures as well, Laurie, or would you just frame it as generalized, like just general gestures?
Right. So we would have, I think, to show the parents just so they can, kind of decide to have a truly informed consent, but it could be modified so that instead of having this persistent randomization, right, where they're randomized to model month opening or term protrusion, and then they stay this way if they participate multiple times. Then each time it's kind of a new one, right? Although if they participate more than 2 times, then there's going to be a repeat, right? So, and we did find that to get, the more repeats we include, up to about 5, we do raise our inclusion rate. So there's a little bit of a trade-off there. But if we end up including 3 gestures with the finger movements, then maybe having 3 repeats, up to 3 repeats, and then it's a different one each time. Maybe that can be a little bit of a compromise there.
It's possible that your study that you're doing at home is such a clever idea and such a good way of getting a large sample that it does, and I think in your talk you got to this, in some ways it can be thought of as addressing a slightly different point. Like whatever you find in the lab, I mean, even if you find imitation in the lab, you can also find non-imitation in the lab, but suppose even babies imitate in the lab. Tongue protrusion varies by the stimulus. The question is, does that have any implications at home in a real life setting, not with all the experimental controls, but at home? So I have a homogeneous background, a spotlight behind the baby to light up my face. Of course I do, because I want to capture the baby's attention on the face. Whether it's tongue protrusion or mouth opening, that's randomly assigned, so it's not an artifact. I want a visible face. That does, but it's a perfectly great question. Do babies imitate their fathers at home? Do babies imitate their mothers at home in real life? So I just wanted to say, in a way, you could think there's value in doing what you're doing, even though you cannot logically, really reasonably have the mother be blind to what they're doing, especially if she's the model. She's going to remember the next day. So But you're asking a slightly different question that's clever enough and important enough that it not necessarily the same thing as how do you make the most sensitive eliciting conditions in a lab where you do have the freedom to design an experiment in an artifact-free way that maximizes the eliciting conditions for the baby. Those are two forks in the road.
Yeah,
And
no,
many people, by the way, if I could just add one other thing, many people are doing studies of early imitation using clinical methods that I don't use. I'll use laboratory experiments with timed intervals in a structured experiment. There are, as Virginia and people know from reviewing the literature, many people out there using more clinical methods where they're putting out the tongue only when the baby looks at them and only so long as the baby looks. And then the baby can look away for 5 minutes and come back, and then they do more. It's not a timed experiment. Many of them find positive results for imitation. There are replications, conceptual replications of those people using those clinical methods at home. So there could be a strength of that method. Not everybody has to do an experiment. I did it with these strictly timed things, but there are other people finding imitation with more clinical methods, but they can be questions. So.
Yeah, I guess, sorry, I didn't, should have specified that I was assuming that if there was an online component to this effort, it would be using, videos or something, or at least if it's synchronous, an experiment on Zoom, something like that, right? But that it wouldn't be the caregiver movements, because I agree that it's a different question, right? Particularly if we're using, baselines and so on, right? We're looking at something different. Now, as far as carryover, having had those people do the same study over and over, I think actually, well, I do, you do get to worry about carryover, but in some way, it can actually be interesting, right? Because you also get to look at learning and again, it has it resonates with some theoretical questions, right? Like if we think it's just reflexes and kind of random arousal, like we shouldn't see any changes, right? Unless like somehow we find just fine-tuning the arousal so that you get the behaviors that match your theory. So and if there is motor learning in those very, very young babies, in order to match a gesture that is being made, I think that would actually be pretty interesting to know. But again, I maybe there are too many confounding potential explanations that we want to minimize that.
This might be beyond the scope of what we can do here, but wouldn't it be so cool if we could invite families to come in for a laboratory visit, but also then invite them to the more standardized online, with a standard video that everybody sees the same stimulus, and be able to look at the correspondence between both? Because I don't know about you guys, but a related issue I have is even if we find these null results at the group level, I'm still going to be wondering if there's meaningful individual differences there, right? Like, could there still be something there and we're just not seeing it when we collapse across the entire group and just look at the average? And I'm going to be presenting some results in our next group meeting kind of that speak to that issue and the need to consider individual differences. And maybe this could give us an opportunity if we had two studies kind of running in parallel and invited people to do both. If you come into the lab, you probably would do an easy, quick little online study too. That's a smaller ask compared to coming in for a lab-based study. I wonder if we could look at kind of how much, if the babies who imitate in one setting also imitate in the other setting, do we see kind of that stability of those individual differences?
Yeah, I think that's a really good point. And I'm just going to jump in here and say, I think, so Liz brought up the fact that she'll be presenting at our next workshop, which will be on the 28th, in the middle of the night for many of us, but at a nice time for people in Europe and for the Australian folks who will be able to make both sessions. And I just want to say that, I mean, this has been a great discussion. These are exactly the type of questions that get tackled in ManyBabies projects. And it's fun to be able to have these debates and come to a group consensus about how to test these things. And I want to reiterate something that I said at the beginning about from the first ManyBabies project, where we had the main project that has spawned a bunch of spin-offs, some of which sort of happened concurrently and some of which are happening well after the fact. So some of these things you've been talking about can, you can have the main project, and then you can also have sort of supplemental spin-off projects that some subset of participating labs do, depending on capabilities and recruitment, depending on how many babies they're able to get, or if they have the capacity to bring people back multiple times as opposed to just once, that type of thing. So there's lots of possibilities there. And there will be plenty of room for discussion of these topics, some of which I'm sure will happen in the next workshop, and then in the planning and design meetings to follow. So I think we can end there unless anybody has anything that they're really dying to say before we sign off.
I'll just go ahead. Just from a process perspective, just to give you a sense of kind of what to expect, probably, although these things flex with different groups, which is that for each of these issues, whether it be kind of age sampling or car seats or carryovers, what groups typically do is they line up an agenda of the key decision points. So you start writing the protocol, and here are the things we're going to have to decide as a group. Okay, some of these are really going to require kind of plenary discussion, and everybody gets together on a fixed schedule every couple weeks or so forth, and then decides, okay, have we made a decision on this, or what further action do we need to take? So you move towards a kind of regular planning schedule where the leadership team is setting an agenda, bringing it to the group, and having that kind of fuller discussion. And we can talk a little bit, with the sort of current leads and kind of any folks who are interested in being more deeply involved, how to go about setting that schedule, and at what point to get the kind of community call for participation in the planning meetings. But that's the general idea, is we're setting some of the agenda items now, and then you work this out and try to figure out what the process is for getting data on the questions, whether it's power simulations or literature review or so forth.
Yes, great points. Yes, so we're not going to solve any or all of those issues today, but this has been a really excellent kickoff for the project. I've really been excited to hear about all of your work and to sort of listen in on this discussion. This is going to be a very exciting project, and I can't wait to hear from our next group of experts in the next workshop, and then hopefully we can get, we'll be recording that session as well, so you'll all be able to see what is shared then, and they'll all be able to view this session, and then we'll move on to the next. We'll get everybody together in one or more meetings to move on to the next step, just like Mike said. So I really want to thank everyone for coming. These projects are only as good as the people who participate in them, and this is really such an excellent group of people. The enthusiasm for this has been outstanding. So thanks everyone for participating, and we'll look forward to the next steps. And also thanks to the audience for coming. With that, we'll say goodbye and see you at the next workshop on July 28th. And feel free to pass that invite around to people as well. All right, thanks everyone.
Bye.